It's really my pleasure to invite the first speaker, who is Francesco Amato. Francesco is a PhD student at the University of Glasgow and is finishing his PhD program now. And over the last 3-4 years, he has put up a nice program on the role of monocytes in modulating chemotherapy response. And he will give us an update today. Thank you, Francesco.
Hi everyone, good afternoon. I want to thank the organizers and the MMF for this possibility to speak about my research today. So I'm going to talk about my research project over the last 4 years and with title Monocytes Contribution to Chemoresistance in Biliary Tract Cancer. I'll be really quick on this part, especially on the background, considering the content of the conference and the background of everyone here. So we know that at the moment biliary tract cancer represents a socioeconomic problem due to the increasing incidence over the last decade. Currently, for patients, chemotherapy represents the cornerstone treatment for biliary tract cancer. And due to the chemoresistant nature of these malignancies, we know that at some point patients will develop resistance. However, we know that there is a small niche of patients benefiting of chemotherapy that is alive at 3 years. So there is an unmet need at the moment just to understand what are the mechanisms that are associated with long-term benefit to chemotherapy. At the beginning of this year, we published a paper in which we characterized the whole transcriptome profile of the pretreatment liver biopsy in a cohort of intrahepatic cholangiocarcinoma patients with extreme divergent outcome to chemotherapy, classified as rapid progressor and long survivors. We identified a signature associated with response, and we investigated this signature applying a digital cytometry approach. And we found out that the cancer compartment, the tumor-associated myeloid compartment, were the main compartments where gene deletion occurred. To better understand the involvement of the tumor-associated myeloid compartment, we applied our signature to single-cell RNA-seq data from resected tissues from a cohort of biliary tract cancer patients undergoing adjuvant chemotherapy upon resection. We found out that peripheral monocytes were the main population of immune cells, and the application of this signature of response could differentiate patients responding from patients with long-term benefit to adjuvant chemotherapy from patients with short-term benefit from benefit from chemotherapy. So my hypothesis was that monocytes may be already deregulated at the level of periphery, and once they reach the tissue, they may alter chemotherapy response. Within this, to investigate this hypothesis, I set up a longitudinal study consisting of collection of bloods from a cohort of biliary tract cancer patients from baseline to time of progressive disease. Every blood was processed in order to isolate the monocytes, and within this study I set up 3 aims. With aim number 1, I aim to study the dynamics in terms of numerical change of monocytes and how this correlated with clinical outcome of patients. With aim number 2, I aim to study the role of monocytes in affecting chemotherapy response on cancer cells. And with aim number 3, I investigated the transcriptomic evolution of these monocytes longitudinally over the course of treatment of these patients, just to understand if there are possible candidates that can be associated with response, that can be targeted. Aim number 3 was part of the project that was funded by MMF. So I investigated, going to the method section, I investigated a prosthetic cohort, part of the REGBIL trial in Scotland, of biliary tract cancer patients selected according to 2 criteria: inoperable disease and administration of platinum combination. For every patient, I collected bloods longitudinally from baseline to time of progressive disease, going through response, and every blood was processed in order to isolate the monocytes that were used for aim number 1, just to study the dynamics in terms of numerical change, and aim number 2, specifically within this contest, I set up two platforms to study the effect of monocytes on cancer cells. So specifically, I developed a model, a two-dimensional co-culture model consisting of culturing GFP-positive biliary tract cancer cells alone or with stained monocytes. Co-culture and monoculture were treated with vehicle and chemotherapy, and biliary tract cancer cells were followed by development of an algorithm that allowed me to track just the biliary tract cancer cells or just to evaluate the role of chemotherapy on them. I developed a model of further complexity represented by a three-dimensional model of co-culture consisting of culturing GFP-positive biliary tract cancer spheroids alone or with stained monocytes. And monoculture and co-culture were treated at the same way as the two-dimensional model. And in this case, I adapted a different method just to track the role of chemotherapy on the effect of chemotherapy in monoculture and co-culture by time-lapse incucyte. For AIM-MAMDER3, I simply isolated the monocytes and then I collected the RNA from this monocyte that was later submitted for RNA sequencing. Going to the results section, my cohort consisted of 50 biliary tract cancer patients that were enrolled from January 2021 to July 2023. As you can see in the plot in the middle, my cohort covers the different subtypes of biliary tract cancer. And for all the patients up to now that are part of this cohort, as indicated by the longitudinal bars, progressed. And for all the patients, I was able to collect multiple samples longitudinally to cover the different stages of the chemotherapy course. And in terms of clinical parameters, our cohort had a time to progression of 7.5 months and an overall survival of 11.4 months. Numbers that are in agreement with the numbers from the cohort of the main clinical trials used as reference at the moment, such as ABC02 and TOPAZ-1. So with aim number one, I wanted to investigate the dynamics behind the numerical change of monocytes and if there were correlation with the clinical outcome of patients. So at the beginning, I focused on the number of baseline. And as you can see on the left part of this slide, when I took into account three parameters of response that were the response rate, the time to progression, and the median duration of response, I didn't see any difference between patients doing better in the clinical setting compared to patients with a poor clinical outcome. However, we know that over the course of chemotherapy, patients go through response assessment in which the clinical benefit is assessed and the best response is recorded. The situation here became a bit different because patients with short chemotherapy benefits had an increase at time of best response in the monocytes count. According to my hypothesis, monocytes may be involved, may play a role in chemoresistance. So I focused on the potential numerical change longitudinally from baseline to time of progressive disease. And here the situation was intriguing because I noticed that there was an increase from baseline in the whole cohort of the number of monocytes. However, this increase was not significant. When I grouped patients based on the time to progression, patients with a time to progression less than 8 months, so patients with a short chemotherapy benefit, had a significant increase of monocytes compared to the patients with time to progression less than 8 months. We know that we have mainly three populations of monocytes: classical, intermediate, and non-classical, with the classical being the source of the tumor-associated macrophages, a population of immune cells associated with chemoresistance. To investigate if there was a shift in one of the three populations, I gave a look to the percentages longitudinally from time of clinical benefit to time of progressive disease. And I did not identify any significant difference from time of clinical benefit to time of progressive disease in all the 3 populations. So this suggested that we needed a more in-depth way just to understand, to characterize the role of monocytes in affecting chemotherapy response. And at this stage, aim number 2 comes up in my study. So I performed co-culture with cancer cells and monocytes collected from patients. I was able to identify 2 main phenotypes of response to chemotherapy, in vitro chemosensitivity and in vitro chemoresistance, tagged respectively in green and in purple from now on. So the first phenotype of response was similar response of cancer cells in co-culture with monocytes to chemotherapy compared to cancer cells alone, while the second phenotype was characterized by reduction of sensitivity to chemotherapy of cancer cells in co-culture with monocytes compared to cancer cell alone. It was interesting at this stage to notice that when we grouped all the results together, we noticed that we had an enrichment of in vitro chemoresistance events when we performed co-culture with monocytes collected at time of PD. We compared to monocytes collected at time of clinical benefit. Comparing the results in vitro and with the clinical results, we identified a really good concordance rate. However, as you can see, at time of PD, we found out that there were like some cases that were not in agreement with the clinic. We grouped these cases based on the in vitro results, and we found out that patients whose monocytes induced in vitro chemosensitivity when collected at time of PD were actually had a longer benefit to chemotherapy compared to patients whose monocytes induced in vitro chemoresistance when collected at time of PD. The results showed up now are relative to one cell line covering the distal cholangiocarcinoma subtype. However, these results were further validated with 2 additional cell lines covering the intrahepatic and gallbladder carcinoma subtypes. I added a further element of complexity at this stage, represented by the 3D co-culture system, in which I used biliary tract cancer spheroids that, in terms of organization and drug availability, are a bit more complex and more similar to the physiological condition. In this case, I was able to find the 2 in vitro phenotypes that were recapitulated, in vitro chemosensitivity and in vitro chemoresistance. The enrichment of in vitro chemoresistance was observed also in this case, with even a better concordance rate between clinical results and in vitro results. While I was collecting the monocytes, in parallel I was collecting other population of peripheral blood mononuclear cells that I used as control. So in this case, we can see that only in the co-culture performed with monocytes, there was an enrichment of in vitro chemoresistance events at time of progressive disease. And giving a look to the functionality of the monocytes and on the peripheral blood mononuclear cells depleted of monocytes, only the monocytes collected at time of progressive disease were actually able to reduce the effect of chemotherapy on biliary tract cancer spheroids. These results show that we have a functional change over the course of chemotherapy. And I put together all the data for patients for which I was able to collect, to perform full longitudinal collection and full longitudinal co-culture. It was really interesting to notice that co-culture performed with monocytes collected at baseline gave rise to the in vitro chemoresistance phenotype on cancer cells in almost the 50% of the cases. However, we did not find any correlation between the in vitro results and the clinical results. It was interesting at this stage that when we moved to time of clinical benefit, the phenotype observed at baseline was changed in 8 out of 11 cases. This indicated that the functional phenotype in monocytes may arise as a response to chemotherapy. It was, at this stage, we wondered if there is a specific stage at which we have the functional phenotype mediating the in vitro chemoresistance phenotype on cancer cells. So I put together all the cases for which I performed longitudinal co-culture and longitudinal collection, and to understand if there were associated also some dynamics behind this behavior, I put together also the clinical information for each patient. This is a representative case in which we performed co-culture from baseline to time of progressive disease. As you can see, here we can observe the functional phenotype mediating in vitro chemoresistance at baseline that changes over the course of chemotherapy to the functional phenotype mediating in vitro chemosensitivity. In agreement with the clinical results, because at that stage was recorded the response for that patient. So as you can see, with the increase of the tumor markers, we have the change of the functional phenotype, and we started to observe the in vitro chemoresistance events from time to response to time of progressive disease. When we gave a look to the whole cases, however, we noticed that in the majority of cases, in almost all of them, we observed the functional phenotype mediating in vitro chemoresistance at time of progressive disease. These results altogether show that we have a functional rewiring over course of chemotherapy, and to understand if it was also associated a transcriptomic rewiring, we performed RNA sequencing longitudinally within this study. Specifically, within this context, I put together all the cases for which I had samples from baseline to time of progressive disease in order to track a potential shift over the course of chemotherapy for each patient, and to cut down also the variability to do the heterogeneity of our cohort. From baseline to time of progressive disease, I identified 112 genes differentially expressed by differential gene expression analysis, and it was interesting to notice that at time of progressive disease, monocytes became less pro-inflammatory. To shortlist all the candidates that were matching the clinical and in vitro outcome, I performed a second analysis, second differential gene expression analysis, comparing the transcriptome between monocytes inducing in vitro chemosensitivity and monocytes inducing in vitro chemoresistance. I overlapped this analysis with the analysis performed, showed in the previous slide, and we found out 2 genes upregulated from baseline to time of progressive disease, and in monocytes inducing in vitro chemoresistance, and 3 genes downregulated at time of progressive disease, and in monocytes inducing in vitro chemoresistance. Considering that we showed that there is, based on the results from our co-cultures, that we have a functional change from time of clinical benefit to time of progressive disease, we investigated the change in terms of expression of these genes from time of clinical benefit to time of progressive disease. It was interesting to notice that one of the genes that was found downregulated was actually characterized by higher levels at time of clinical benefit with reduced level at time of progressive disease. While for the 2 candidates that were found upregulated, it was interesting to notice that their levels changed from time of clinical benefit to time of progressive disease. So overall, by this study, I was able to observe and to find that peripheral monocytes count derangement is observed over the course of treatment. The addition of monocytes can actually change response to chemotherapy of cancer cell and biliary tract cancer spheroids. And we also observed a longitudinal evolution of the functional phenotype over the course of treatment. And to this functional evolution was also associated a longitudinal transcriptomic rewiring. Finally, by the transcriptomic approach, we were able to identify potential candidates that at this stage deserve to be further investigated, that are associated with response. So I want to spend just a few seconds just to thank all my team from Professor Bracconi Lab, the members, and first of all, patients and families of the Reg Bill cohort, and the people that contributed to this project from the Cancer Research UK Scotland Institute, all the members of the NHS, specifically part of the clinical trial unit that contributed to the collection of these samples and to and the realization of this project, and our amazing collaborators from University of Copenhagen that helped us to perform the transcriptomic analysis to identify the candidates associated with response. Thank you very much.